Hello, welcome to chapter 17. This video is specific to vision. So let's look at vision. Vision is the actual act of being able to see. What we see is this visible light that is found within this electromagnetic radiation. This electromagnetic radiation is waves. These waves that come from the sun. There's lots of different types of rays or waves. We have gamma rays, X-rays, UV rays, visible light, infrared radiation, microwaves, and radio waves. Those are listed in order. We only see this small portion of all of these different types of rays. There's electromagnetic spectrum is the range of this electromagnetic radiation. So that's this wavelength. A wavelength, going into a little bit of physics here, is from one peak to the next peak. So from one to the next, that's one wavelength. They can be very short, they can be very long. We see that visible light, which is a certain wavelength. Here's all of these different rays. We see from this 400 nanometers wavelengths to 700 nanometer wavelengths. And the size of that wavelength determines what color our eyes can see or interpret. So let's start looking at all the different parts to the eye. Our eye is not just the actual eyeball. There's a lot of accessory structures found in and around that orbit in that skull. So we have eyelids, eyelashes, eyebrows. We have a lacrimal apparatus. This is what makes tears. And then we have 6 muscles that are going to attach to the outsides of our eyeballs. And this is what allows you to move your eyes to be able to see and read. So we're going to start by looking at the eyelids. Another name for eyelids is palpebrae. We have an upper and a lower palpebrae with a fissure, which is that opening between the two. Our eyelids have a lot of different features. They're shade your eyes so you can sleep. Darker allows sleep to happen. They also protect from excessive light and foreign objects. If something comes close to your eye, your instinct is to blink or close your eye to protect it. That lacrimal apparatus that secretes tears, those tears are lubricating. They help to lubricate and keep your eye moist. Our upper eyelid does most of the moving. And we have a specific muscle that opens your eye. We talked about in the muscle chapter how we have orbicularis oculi. That is the muscle that closes your eye. I'm pretty sure I mentioned, but we have levator palpebrae superioris. This is the muscle that lets you open your eye. If anybody has ever had a twitch in any of those muscles that are right around your eye, usually they last a few seconds and they're harmless. Stress and fatigue are the most common causes of those muscles at your eyes to twitch. I had mentioned that palpebral fissure, which is that opening between those two eyelids. There's a lateral commissure, which is just the angle of the eye. We have a medial commissure, which again is that inner angle. And then we have a lacrimal caruncle. These are sebaceous glands and sudoriferous glands. These are secreting out material that kind of collects at that medial eye. That's formed from these glands. And if you ever get those little eye crusties in the morning when you first wake up in the corner of your eye, that's from these sebaceous and sudoriferous glands. Your eyelashes are attached to this tarsal plate. Again, we have sebaceous glands, and it's helping to prevent your eyelids from sticking together. A chalazion. This is when that tarsal gland gets infected. It can produce a little cyst on the eyelid. Conjunctiva. The conjunctiva is a protective membrane. It's that non-keratinized stratified squamous epithelium, and then there's goblet cells in there. So we have a palpebral conjunctiva and a bulbar conjunctiva. The bulbar conjunctiva is that vascular layer. There's lots of blood vessels found throughout there. So if somebody has bloodshot eyes, those blood vessels in that bulbar conjunctiva have dilated, they have enlarged. And that is what can cause someone to have bloodshot eyes. So let's look at an eye. So that medial commissure that I mentioned is that middle corner of the eye here. The lateral commissure, it's found at that outside of that eye. And see that upper eyelid, that lower eyelid. The conjunctiva sits over top of the sclera. So that conjunctiva is a clear layer over top that helps to kind of protect that eyeball. It's got all those blood vessels in it that can give that bloodshot appearance if you didn't get enough sleep. If you're coughing a lot, it can actually cause the blood vessels in your eyes to dilate, and you can get some pretty bloodshot eyes. That palpebral fissure, that opening in the eye, and I hope we all know what eyebrows and eyelashes are. So, what function do our eyebrows and eyelashes have? They are supposed to protect the eyeball from foreign objects, perspiration, and direct rays of sun. I mean, eyebrows aren't necessarily that big to protect against those direct rays of sun, but those eyebrows can catch perspiration. Those eyelashes, as you close your eye, blink as something may come close to your eye, those are helping definitely to protect that eyeball from something coming at it. We have sebaceous glands at the base of every hair follicle of our eyelashes. They're known as sebaceous ciliary glands. They are lubricating your eyelashes. If one of those sebaceous glands gets infected, that's what causes a stye. This lacrimal apparatus. There's a lot of different parts that make up this lacrimal apparatus, but its main function is to produce and drain tears. So it does a lot for us. So it produces lacrimal fluid. And it is produced in these lacrimal glands. I'm looking for a picture. So here's this lacrimal gland over here. This lacrimal gland is going to make those tears. It gets ejected out onto the surface of the eye through the excretory lacrimal duct. So it comes out at the outer portion of the eye. By blinking washes this fluid over the surface of your eye to help keep your eye moist. It's always producing small amounts of fluid to help keep those eyes moist. Then we have, if you've ever looked real close in the mirror at the corners of your eye before, you can see these little tiny holes. You usually can see the one at the bottom, but these are lacrimal puncta. These lacrimal puncta are draining the tears that are coming from that lacrimal gland. At that point, they are going to either travel through the superior lacrimal canaliculus or this inferior lacrimal canaliculus. So they're going to travel up or down. Then we have this excremal or excretory lacrimal sac. This excretory lacrimal sac is where those tears are going to drain. And from there, they're going to travel through this excretory nasolacrimal duct into the nasal cavity. This is what causes when you cry your nose to run. Because, yeah, some of these tears are coming off the face, but they're also being drained into this lacrimal sac, which drains into your nasal cavity. The lacrimal gland is supplied by parasympathetic fibers that are on the facial nerve. If that lacrimal sac gets infected, the cryocystitis, it can result in blockage of the nasolacrimal ducts. That's that lacrimal sac found on the inner side that drains into that nasal cavity. So, as I went through in the picture, but the lacrimal gland will secrete tears into the lacrimal ducts, which will, as you blink, it'll get distributed over that eyeball, which will go through that inferior, superior lacrimal canaliculi into the lacrimal sac through the nasal lacrimal duct into the nasal cavity. That is the flow of tears and how it happens. So now let's look at the muscles that attach to your eye. So first off, in our eyes, or in that orbit in the skull, there's a lot of periorbital fat. Your eyeballs sit in a pretty good cushion of fat. This is helping to protect that eyeball. And then coming through that fat adipose tissue, we have 6 muscles. Superior rectus, rectus means straight, so it's coming straight out on the top. We have inferior rectus, lateral rectus, medial rectus, we have superior oblique, and inferior oblique. We have 3 cranial nerves that go to those 6 muscles: oculomotor, trochlear, and abducens. Trochlear only goes to one nerve, should be lateral rectus, and abducens goes to one muscle. Trochlear goes to one muscle, not nerve. Trochlear goes to one muscle, and abducens goes to one muscle. Oculomotor gets the rest. So those 6 muscles allows you to look all kinds of directions. If you think about the last time you went to your eye doctor, one of the ways they check these 6 muscles to make sure they are working is they have you follow their finger, follow some sort of object around, moving your eyeballs around. This is allowing them to make sure these muscles are working correctly. So those rectus muscles let you look like superior rectus lets you look straight up, inferior rectus lets you look straight down, lateral rectus lets you look out, medial rectus lets you look towards your nose. Those oblique muscles allow for some rotation, like rotary motion. So like up and out, down and in. So you're able to move your eyes in more directions. So let's look at some of these muscles. So here we have that superior oblique. This muscle is coming straight in. Same with that inferior rectus. Here's that inferior oblique muscle here that is coming across underneath that eyeball. Superior rectus is going to do some of that same movement, like coming across the side. They don't show a good picture of any other muscle here for some odd reason. So you can see, we'll be able to see them all in the lab. There's plenty of models in the lab that show all of these specific muscles. So let's look into the actual eyeball. Our eyeball has a very superficial layer that has the cornea in the front and the sclera in the posterior. So the cornea is clear. It is this transparent part over the eye. The cornea can get oxygen from the outside. The sclera is known as the white of the eye. That sclera also helps to give that eyeball its shape and allows muscles to attach. So our eyeball is made of different layers. The next layer in is known as the vascular layer. The vascular layer, that middle layer, has choroid. It's very vascularized, so it has lots and lots of blood vessels in there. We also have melanocytes, those pigment producing cells that we talked about with the skin and your hair color. We have melanocytes in there. They produce melanin for that eye, which makes that layer look dark brown because you're seeing all that melanin that's been produced. What this choroid does is this melanin, as we talked about in the skin, how it absorbs light. Well, it's going to absorb light rays. So it's preventing damage to other cells from light by absorbing that light rays. Then we have the ciliary body. This is the anterior portion of that layer. In there, we have the ora serrata, ciliary processes, and this ciliary muscle. The ciliary muscle holds on to these ciliary processes, which are attached to suspensory ligaments that are going to help attach to the lens, and then the ciliary muscle helps to change the shape of the lens. The iris is the colored portion of your eye. So when somebody asks what your eye color is, they're asking what color your iris is. The iris, again, has melanocytes, which is what gives that specific color, but it has muscles within the layer of the iris because these muscles can change the shape of the pupil, which is the opening in the eye that allows light to enter the back of your eyeball. As I mentioned, the melanin gives your eye its specific color. But that pupil, back to the pupil, it appears black because light's getting shined into the back of your eye. It's dark back there. That's why it looks black. So if a bright light is directed directly at that pupil, as it reflects off, it gives this red kind of appearance. The two muscles within the iris that change the size of the pupil, we've got sphincter pupillae, which is going to constrict. Think of a sphincter as small and tight. So it's going to constrict when you're in bright sunlight or bright light. Dilator pupillae dilates. So when it's darker, your pupils usually are larger to allow more light to enter so you can see better. When it's so bright outside, those pupils constrict because you don't need as much light going in. So let's look at what we've talked about so far. So that sclera is that white of your eye, makes up most of the outside layer. Then we have that choroid, which is that middle layer. It has lots and lots of melanocytes in there because it's producing that melanin that's going to help kind of, really helps to absorb light rays. This ora serrata is this portion that kind of travels all the way around the eye. We have this ciliary body with these ciliary muscles and the ciliary processes. Those ciliary processes are holding those suspensory ligaments, which are also known as zonular fibers. They are what attach, to the lens. That iris is this colored portion of your eye, and then the pupil is this opening in your eye. We also mentioned the cornea. The cornea is this clear layer on your eye. It's on the visible portion of your eye. If you wear contacts, you're putting contacts on top of that cornea. Here, you can see the iris, the pupil, And then you see all of these ciliary processes that would be holding that help or attaching to those suspensory ligaments that is holding that lens in place. We talked about those different muscles. So sphincter pupillae, it constricts when there's bright light. We dilator pupillae, it makes the pupil larger in that dim light. So now let's go into the very inside layer. The inside layer is known as the retina. The retina contains the beginning of the visual pathway. So there's photoreceptors that were mentioned before, or may have been. Photoreceptors are the special receptors used by the eye to allow us to see. These photoreceptors are found in the retina. An eye doctor can use an ophthalmoscope to view the retina. And in the back of the retina, we have this optic disc. This optic disc is considered a blind spot. There's no photoreceptors, no sensory receptors found there. This is where that optic nerve leaves the eyeball. So if I come back up, so here you can see this optic nerve. So this area where this optic nerve is leaving the eyeball, that is the blind spot. So nothing, if light is shined or shined directly on this blind spot here, we will not see anything. And then we have the retina as that, most inner layer of this eyeball here. So this whole inside that is shown in yellow here, that is the retina. Here's what the inside of the eye looks like when using an ophthalmoscope. Those of you going into nursing may actually get to try one of these. But here you can see the optic disc. It's a little bit lighter in shade than the rest of the retina. There's also this macula. And in the middle of the macula, we have this fovea centralis. This is the area in the back of the eye that has the most precise vision. This is where we see the best. So when something is reflected directly into this fovea centralis, that is where we see the best. So sometimes you might catch yourself tilting your head, moving your head different directions to try to focus on something. That's focusing whatever image or whatever you're looking at into that fovea centralis so you can see it the best. So in this retina, we're going to have a pigmented layer again, because we've got more melanin containing cells, because we need help absorbing light rays. Then we also have the neural layer. This contains the different types of neurons that are going to allow us to see. So the photoreceptors, this is the layer that is picking up the images of what we're seeing. The bipolar cell layer, this is a connection layer that's going to connect to the ganglion layer. This ganglion layer is the layer that connects to that optic nerve. makes up the axons of that optic nerve. So in the retina, we also will find horizontal cells and amacrine cells. These form some lateral neural circuits. These can modify signals as these different images and everything is passed along these cells. So when light gets passed along, they go through the ganglion layer, the bipolar layer, before they reach that photoreceptor layer. So for images to be picked up, they have to travel through these layers, and then it kind of travels backwards back out. We'll look at some pictures of that in a minute. But we've got two different types of photoreceptors in our eye. Rods. Rods are used for dim light. They do not have any color vision. So if you've ever gone out or gotten up in the middle of the night to go to the bathroom, you don't turn on any lights while you're on your way. When you're trying to see, you kind of notice that everything appears kind of black and white because all that's picking it up is rods. There may be some shades of gray in there as well. Your book uses the example of the moonlight. The other type of photoreceptors are cones. Cones color. So this is where we get our color vision. Rods and cones are test questions. Cones are also going to pick up that brighter light. We have blue cones for blue light. We have green cones for green light. Then we also have red cones that pick up red light. Everything that we see that is outside of this blue, green, or red are combinations. There are all these different combinations that allow us to see these different colors. So most of what we see goes through cones. If you lose cone function, it actually gives legal blindness. The loss of rods just means you have difficulty seeing in dim light, and these people should not be driving at night. So age-related macular degeneration or macular degeneration. This is just a degeneration of that retina that happens in people 50 years and older. So there's different abnormalities that happen within these different regions of this retina. And it's usually in that area of most acute vision, that fovea centralis that I showed. People can retain their peripheral vision because most of your peripheral vision is cones. They lose their ability to see straight ahead because that's where most of those, or no, peripheral vision is rods. Rods is more dim light. Cones are more straight back. Sorry, my bad, said it backwards. This is actually the leading cause of blindness in those over 75, is this age-related macular degeneration. In those who smoke a pack a day, it's more common. Again, smoking is going to affect everything somehow in your body. The visual field usually starts out with blurring and distorting, and people usually go seek out help when all of a sudden they can't see things quite clearly. So here's these different layers within this retina. So light is traveling this way. That is where these cones or rods are going to pick up whatever has just been projected onto that eye. From there, this information is going to travel through that cone or through that rod, and it's going to attach to this horizontal cell, bipolar cell, or amacrine cell. This is considered that bipolar layer. So these cells, they could connect directly to these bipolar cells. They could connect to these horizontal cells, which will then connect maybe to an amacrine cell, which will go down to this ganglion cell layer. If you kind of noticed these cones here, they attach directly to bipolar cells, which go directly to horizontal cells. These rods, they are going to attach to these horizontal cells. I think I said that wrong too. Cones attach directly to bipolar cells, which go to the ganglion cells. Rods will attach to horizontal cells first before they transfer over to these ganglion cells, which then form those axons of that optic nerve. There we go. And the reason that happens, our cones are going to connect directly to these bipolar cells because cones provide the most precise vision. This most precise information is traveling through these bipolar cells right away. Cone vision isn't as clear, and it's evident because you can see this one cone here, or this one horizontal cell is attached to 1, 2, 3, 4, maybe 5 different cones, rods. I keep saying those backwards. They attach to all these different rods, one horizontal cell, which is why our vision is not as clear in dim or dark light versus those cones go directly to those bipolar cells, which go directly to those ganglion cells, which provide a lot clearer, more precise vision. So as these cells come down, these rods, these cones, they're coming and they're connecting through this ganglion layer. All of these axons, what I wanted, come through and form this optic nerve. This is traveling back to that occipital lobe, which has that visual field in it. So here's a slide of all of these different layers. You can see all of the different cell bodies that are found in those specific cell layers. You see all those little dots, those are cell bodies. Even that ganglion layer, you see those cell bodies. And then those synaptic layers, you can see some of those axons going through. Here, we can see it in an electron micrograph. So you see all these cones that are kind of in white, or those rods in white, those cones in green. Knowing my shapes today seems not to be working. I'm trying my best. So this shows the layers and how everything passes through the retina. Photoreceptors, outer synaptic layer, bipolar cells, intersynaptic layer, and ganglion cells. Those ganglion cells form that optic nerve. I mentioned that blind spot where that optic disc is. There's no rods or no cones. I mentioned that macula. This is on the back of that retina. And in the middle, we have that fovea centralis, as I mentioned. This contains only cones. Is that because I said it was the most precise vision we have? Cones give us that most precise vision. So it's that area of highest visual acuity. And then I mentioned we tilt our head around because we're trying to focus into that fovea centralis. We have more rods in the periphery. So if you tilt your head to the side more, you may see stars better because it's black and white in that sky. A detached retina, something interesting. Some of this is age related, but it could be because of trauma to that eye. Somebody punched you in the head, or that just age related. That retina layer kind of comes detached and falls over and kind of folds over on itself. I've heard people describe it as closing, they feel like a curtain is closing over their eye. Like they can visually see, and as that retina kind of folds over, a curtain is closing and everything goes black. Because there's fluid in the back of our eye, this fluid starts to accumulate between these layers. And only way you can put it back is through surgery or cryosurgery. Cryo is cold. And these people become blind in that eye until it becomes attached. So we mentioned the lens. The lens is behind the pupil and that iris. It is made of proteins called crystallins. Test question. It is arranged in layers like an onion. There's no blood vessels that go to the lens. It is held in place with those zonular fibers, those suspensory ligaments that attach to those ciliary processes that I was mentioning before. Your lens's job is to help focus images onto the retina. Just like lenses in glasses, your lens is a natural form of like a glasses that we have that helps to focus in. So here's that lens. As we can see here, I'm coloring in red highlighter. This lens is made of that crystalline protein that it can change shape. So let's look at how our eye is divided up. Our eye is divided into an anterior and posterior segment. The anterior segment is between the cornea and the lens. So that clear layer on the front, all the way through to the lens. The anterior chamber is between the cornea and the iris. And then we have a posterior chamber that is behind the iris in front of those zonular fibers and lens. This area specifically is filled with aqueous humor. Test question. Aqueous humor is going to nourish the lens and the cornea, so that front part of the eye. This drains out. Let's look. We have blood vessels in part of the ciliary body here. From those blood vessels, we're pulling out fluid and we are making aqueous humor. This aqueous humor comes out of these blood vessels. It's going to go into that posterior chamber of the eye. It's going to come around, going to go in front of that iris to that anterior chamber, imagine that posterior chamber, where here it's going to go and drain into that scleral venous sinus. So this fluid is being produced and drained every day, all day, every day. That posterior segment, this is between the lens and the retina. This is filled with more of like a jelly-like substance that is known as vitreous humor. This vitreous humor helps to maintain the shape of the eyeball. It fills about 4/5 of the eyeball because the back of your eyeball is actually larger than the front portion. This does not get replaced like the aqueous humor in the front. The vitreous humor, this is another test question, is the vitreous humor in the back portion of the eye, also known, can be called the vitreous chamber. It's formed in embryologic life and it stays there your entire life. There is phagocytic cells in there because it helps to remove out debris. Once in a while, some of this debris can cast kind of a shadow on that retina, and you can actually kind of see it. Those are known as vitreal floaters. If you've ever watched Family Guy, there is a entire episode of Family Guy about vitreal floaters, and he calls them by name. He's named them. It's more common in older individuals just because their immune system does not work as well. So those phagocytic cells are not necessarily cleaning out any debris like they should anymore. So they're harmless. So our intraocular pressure. So that pressure inside of your eyeball should sit at about 16 millimeters of mercury. That aqueous humor in the front and that vitreous humor in the back is helping to create this intraocular pressure. Why? We need to maintain the shape of our eyeball. So this chart is kind of nice. It goes through and it kind of highlights the different parts of the eyeball for you so you can see where everything is labeled and highlighted on all of these structures. That lens and what it does, the functions of it too. Anterior chamber, posterior chamber. So let's look at how we see. We mentioned those light rays. Those light rays are going to be refracted, which is bending of that light. The first place that those light rays get refracted is that cornea. That cornea is going to bend that light to focus it onto the lens. The lens will refract that light to focus it onto the retina. Here's the fun part. When an image comes in, this is what we normally see. This is what is refracted or shown, focused onto that retina. So it's upside down and it'll go through that right to left reversal. So everything is going to be flipped right to left and inverted upside down. The brain takes this and is able to flip it back and invert it so it's now right side up, so we can know what we're looking at, even though what is reflected onto our eye is completely different. So most of the light refraction happens at the cornea. Only about 25% of it happens at the lens. So when something is about 20 feet away, light rays are coming in at almost parallel. The lens has to bend these parallel rays so they are focused into that fovea centralis. When something is closer than 6 meters or 20 feet, those rays have to be refracted even more. So they are focused into that fovea centralis. So the best example of how this is done is light rays being refracted through water. This is the best, like, actual experiment that can be done that shows how these light rays are being refracted just like they are on our cornea and then through our lens. So your lens is able to change shape. So a surface that is curved outward is known as convex. So, and it has that outward curve to it. Surfaces that are inward curved is known as concave. So when something is convex, that lens will refract light towards each other so they intersect. But if something is concave, light rays refract away from each other. So if the lens is convex on both sides, its focusing power is better. It's a lot greater. When focusing on a closer object, that lens becomes even more curved. This is accommodation. We increase the curvature of that lens to help us see. Near point vision. This is the minimum distance from the eyes an object can be clearly focused with maximum accommodation. In a young adult, it's the best. It's about 4 inches. They can hold something about 4 inches in front of their face and they can see. As you get older, it becomes further and further away. If you've ever seen your parents or grandparents having to hold something out in front, like very far out in front of them so they can see, can read something, their near point of vision has grown tremendously with age. So when we view something that's far away, those ciliary muscles relax. When that happens, the lens flattens out. When something is close, that ciliary muscle has to contract. It helps to pull this lens forward, releases tension on the lens and the zonular fibers. That lens becomes even more convex. We have parasympathetic fibers from the oculomotor nerve that are going to those ciliary muscles to allow for that accommodation of that lens. So a distant object, as it's coming in, that lens and that cornea work to focus onto that fovea centralis. When something is close, that lens becomes more convex to allow us to be able to focus something onto the back of the retina. Presbyopia. As I mentioned, aging, that lens will lose its elasticity and its ability to focus. So by 40, that near point of vision is usually about 8 inches away from their face. By 60, it can be up to 31 inches from their face. So people having to hold things at an arm lengths out. It begins in their 40s, mid 40s. People start needing glasses for reading or bifocals if they're already wearing glasses. Around this time, just because of that lens losing its elasticity. So an emmetropic eye, this is normal. They can refract light rays clearly onto the retina. Myopia, nearsightedness. Somebody who has nearsightedness, their eyeball is too long. So they have problems focusing directly onto the retina. Instead, images get reflected in front of the retina because that eyeball is too long. They can see close objects, but they cannot see distant objects. Nearsighted because that's what they see. Hyperopia or farsightedness. These people, the eyeball is too short. So that image is reflected, comes together behind the retina. They can see at a distance, but they cannot see close. So it's named nearsightedness and farsightedness depending on what they can actually see. Nearsightedness can see near, close. Farsightedness can see far away. The other one, an astigmatism. Either the cornea or the lens has an irregular curvature to it. So some of that image may be out of focus. For those of you who have an astigmatism, here's something fun. People with an astigmatism can either see things that are straight up and down, clear, or they can see something that is horizontal, clear, without corrective lenses. So it's kind of fun to go out without your glasses or contacts, corrective lenses, and just look at around you. And you'll see, like me, oh, I can see those tree trunks that are straight. Perfect. Everything else around it's a massive blur, but those straight up and down as clear as can be. Somebody else, it may be something that is horizontal, that is clear as day. So everybody is different, and it's kind of cool how one person sees one thing while somebody else sees another, and it just has to do with the way that lens or cornea is curved differently. So this shows that nearsightedness, they can see things close up. Normally, that image is focused in front of this retina back here. So they have to use a concave lens to focus that image to the back of the eye. Well, farsightedness, they can see far away. Their eyeball is too short, so that image is focused behind that retina. They use a convex lens to focus onto the eye. So most things can be corrected by glasses, contact lenses, or surgical procedures. Contact lenses actually float on a film of tears. They don't sit directly on that cornea. They actually float on this film of tears that's covering that eye. LASIK, they are reshaping the cornea to correct refraction problems. The problem with LASIK is the aging that happens to the eye is still going to happen. I've had uncles that have had LASIKs that are back in glasses again as they've gotten older. So it works while you're young, but those normal effects of that lens losing its elasticity is still going to come in and take place. So constriction of the pupil, that is that narrowing of that hole where light enters the eye. It's an autonomic reflex. What we're trying to do is prevent too many of those light rays from entering to the back of the eye. So that pupil will constrict to bright light. Binocular vision. By both of your eyes focusing on something, this is what allows that depth perception and for you to see objects in a three-dimensional shape. So those light rays that hit that from that object come into both of those retinas. You're right, both eyes. And as that image comes in and is reflected off of, or onto those retinas, because of both of them, it allows you to see that depth of that specific object. So as we move closer to that object, the eyes have to rotate medially if those light rays from the object are there to kind of strike the same point on those retina. Convergence. Convergence is the medial movement of both eyeballs. This is something that, again, eye doctors sometimes will check. They hold something between both eyes, move it closer to your nose, and you're to check how your eyes come together, because those muscles that attach to the outside of your eye allow for that convergence. So our photoreceptor function. So that outer layer is of that retina is rods and cones. Rods, dark vision, cones, color. They are going to pick up photopigments. They are using these integral proteins that are in the membrane of these photoreceptors to help pick up these different colors. So those cones, they're going to pick up, we have blue, green, and red cones. So they allow us to pick up those colors, and combinations of them let us see different colors. Rods, they have rhodopsin. That rhodopsin is the photopigment found in rods. This rhodopsin looks like a stack of coins. There's this stack of these many rhodopsin photopigments. These photoreceptors, they're renewed very quickly. Rods, 1 to 3 new discs are added every hour as the old ones at the top shut off. So you can kind of see these discs in here of these rods. New ones are being made down here at the bottom, and they work as new ones are being made, they work their way up to the top where some of them get pinched off. There we go. If you're colorblind or know somebody who is colorblind, color blindness is inherited. And it's an inability to distinguish between two colors. So they usually have a deficiency of one of the type of cones, that red, green, or blue cone. But the most common type of color blindness is red-green color blindness. In red-green color blindness, either the red or the green cones are missing. So they can't tell the difference between that red and that green. Night blindness. Night blindness is not being able to see well in low light. And that has to do with your rods. Possible vitamin A deficiency. We need vitamin A to make rhodopsin. So these photopigments, we have opsin, which is a glycoprotein, and retinol, which comes from that vitamin A specifically. So to maintain good vision, you need good vitamin A. Here are some different foods that contain vitamin A. Or liver. So our photoreceptors function. They're going to go through this isomerization. I can't say that word today. So in darkness, retinol has this bent shape to it. This is this retinol. You can see this bend in it. In darkness, it has this bent shape. When light hits this retinol, it changes its shape. That's that isomerization of retinol. Is that changing it from this cis shape, which is bent, to this trans shape, which is straight. Then once we get out of the light, that trans shape, it's going to separate from that opsin. That opsin is all of these little spring-like appearances that we see inside of this photoreceptor. So it separates out. When it separates out, what it causes is this bleaching effect. That's why when you see something bright shines in your face at night, that bright light goes away, but you can still kind of see that light for a little bit. That's bleaching. So by bleaching of this retinol, of that opsin, retinol leaves, it's going to go from that trans straight line to that cis bent line, where it is going to come back in and bind with that opsin again, that regeneration. That is what is happening inside of our rods. So that light adaptation, that occurs in seconds to that brighter environment by decreasing that sensitivity. But that dark adaptation, so when you enter a dark room, That would happen slower. So in in darkness, full regeneration of these photopigments happens usually in the first 8 minutes. So in daylight, regeneration of opsin cannot keep up with the bleaching process, so rods they contribute very little to what you see during the day. Cones regenerate very rapidly. That some of those cis forms are always present, even in very bright light. So this is what is happening that allows you to see that bright versus dark between these rods and these cones. Phototransduction. What is happening in phototransduction is where that light energy is converted into what'll be that action potential. Got a picture. I like pictures. So we had mentioned how in the eye we use second messengers. We talked about it. We'll talk about it with the ear, or if you've already listened to that lecture, they use it there too. But here, with that cis and that trans retinol found in that rod, what happens is first we start with this cis retinol shape, so it has that bent, which means it's inactive. But once that light hits, it activates this cascade of events, which is eventually through this cascade of events, is going to cause sodium channels elsewhere in this rod to open. Because we have this membrane up here of this rod, this top disc is what is bringing in the light. But what is happening is through this step-by-step process, these many steps, it takes till step 5 for these sodium channels to even open. And it is this cyclic GMP that is being released from this guanylyl cyclase that comes and binds to these sodium channels to cause these sodium channels to open. And what is happening is for a rod, when darkness hits this rod, it will depolarize. When light hits this rod, it's going to hyperpolarize. So it's going to go one way or the other. Depolarize, it's sending that information back into that visual center in the occipital lobe, or it's hyperpolarizing, which is preventing sending off that signal. So this goes through a lot of that. So let's go through the visual pathway. Again, pictures. In this visual pathway, that optic nerve that's leaving the eye comes together with this optic chiasm. This optic chiasm here is this cross. Didn't want to do what I wanted it to do, so I'm going to pick this instead. Where there will be some cross of fibers. From there, we are going through the optic tract, which if we keep following it back, we get to this lateral geniculate nucleus, which is found in the thalamus. And then we have this optic radiation. All of these fibers traveling to that primary visual cortex and that occipital lobe. From there, the information is going to go to the association centers of that occipital lobe, so we know what we're looking at. Going to that primary visual cortex lets us know we're looking at something. When we go to those association centers, that's what lets us know what we're looking at. So here's something kind of interesting. Our visual fields. How things are reflected into the eye. So if you follow the colors, like this dark red, this outside of this left is actually going to the medial port of that left eye. This middle, this over here, is actually going to the outside of that left eye, while this kind of dark yellow goes to the outside of the right eye, and this outer area of that field is going to the inside of that right eye. This is how things are reflected into the eye. Then it travels back on this optic nerve. Now we are taking both outer ends are crossing over. So what we are seeing in this visual field is going to travel back to the opposite side of the brain. What we're seeing on the left side of the eye is going through to the right side of the brain. So what we're seeing is going to the opposite side of the brain even. So this kind of breaks it down a little more to show these different fields of vision and how they're going back to different areas. So this visual field is helping to create this binocular vision by these visual fields that overlap a little bit. So light rays from this object in the nasal half fall on the temporal half of the retina, and vice versa. Visual information from the right half of the visual field is conveyed to the left side of the brain, and vice versa. So this goes through this whole visual pathway that I showed in the picture up above, showing that information coming in goes through that optic disc, goes through that optic chiasm. Some of these fibers are going to cross over. They're going to go through the thalamus, that geniculate nucleus, and then they're going to go all the way back to that primary visual cortex of the occipital lobe. And that is everything on vision. Thank you very much for listening, and have a wonderful day.